Good morning, everyone. Good afternoon for those that are some other part of the world. My name is Samhwas Wonkam. I'm a medical geneticist working at University of Cape Town. It will be my pleasure and privilege to chair this session this morning. We have three excellent presenters, two that will be speaking to science and education, and a third presentation that is promotional from our industry partner. It will be my pleasure and privilege to introduce to you Professor Nicola Mulder. Prof. Nikki is well known by every one of you. She's the emperor, or I don't know how you say it in English, of a big empire, called Bionet, that have connection everywhere in Africa. Believe me, she can speed dial every single institution in Africa regarding bioinformatics. I have the pleasure of being working with her on the sickle cell disease for the past 5 years, developing the sickle cell disease ontology, and now working to coordinate activity in at least 7 different African countries. Today, Nikki will be speaking to us to data science and genomic in Africa. Welcome, Nikki.
Thank you. Share my screen. Okay, so thank you very much to the organizers for the opportunity to speak. So I was asked to talk on data science and genomics in Africa. I'm actually going to talk a little bit more about my area of expertise, which is the bioinformatics side of the data science, and a little bit more specifically about human genomics, although I will mention some of the other genomics towards the end. So data science is really an interdisciplinary field that uses various different methods, algorithms, systems, etc., to work on structured and unstructured data to derive novel insights. And the key components would be data mining, machine learning, and big data. And we see that when all of these require data infrastructure and tools. And if you look at the genomics workflow, the data side of genomics really covers every aspect of the workflow, from the data collection, when you're looking at CRFs collecting data from patients directly, from wearables, environmental data. Then there's data generation from samples, perhaps people doing genome sequencing or other omics experiments or taking measurements on blood. All that data needs to be transferred, curated, stored somewhere. It needs to then be processed, analyzed, and interpreted, and then potentially downstream integration and further knowledge gain to translate that into some real health benefits. So there's a serious data infrastructure that's required to have this working smoothly. We need data transfer protocols, standards for moving and for analyzing data and curating data, storing it securely, making sure if it's human data that it's encrypted and secure and protected, having the amount of compute required to analyze the data, and including obviously the tools and the workflows. And so this all comes with a desperate need for data-related skills. And it's not just the bioinformaticians to analyze the data, it's the IT people, sysadmins to manage the workflows, and the data infrastructure, the computing infrastructure, and then the curators. So what do we have in Africa? So on the data science side, I mean, I'm the first to admit that I haven't really looked at in terms of the broad definition of data science and what there is in Africa. There are various institutes, like there's Data Science Africa, and I've provided the URL there. We have the African Open Science Platform, which is looking at enabling data science and open science in Africa, and that's very broad, from biology to environment to astronomy. And then, of course, many of you be aware of the new NIH initiative, the Data Science in Africa for Health, and that's just the funding's the awards are being made anytime soon. So in bioinformatics, we've seen a huge increase in bioinformatics capacity in Africa, and I think that's largely to H3 Bionet. When we started 9 years ago, I think we were in a very different state to where we are now in terms of what capacity we have. But there are also other initiatives. So the African Society for Bioinformatics and Computational Biology and regional groups of the International Society have all helped to develop bioinformatics awareness and training in Africa. So H3 Bionet is a Pan-African bioinformatics network for H3 Africa. So as Rose said, it's quite widespread. We have 28 partners in 17 countries, more than 200 different members across the continent. And our aim is really to build the infrastructure in terms of the compute, the workflows, the tools, and the skills to enable genomics research in Africa. We've trained more than 4,000 individuals to date on various aspects of data. And we try to encompass all these pieces of the data infrastructure. So in terms of the data infrastructure, the storage and computing, we have built an infrastructure that's up to 40 million. We've used 40 million core compute hours to date. We can store 3 petabytes of data. If we look at the different components around the continent and we put those together, so we have built reasonable infrastructure and also secure data storage and movement. Trying to get over the poor internet we have in many countries. We've also built the infrastructure for storing and archiving African genomics data, and this is based on the infrastructure that's the architecture of the European Genome Phenome Archive at the European Bioinformatics Institute. So we've really tried to model it on a robust system to ensure safe, secure transfer of data, storage of data, and access to data. And in doing so, we've trained systems administrators and bioinformaticians in order to be able to set up this infrastructure and run it effectively. We've also done a lot of work in terms of data management, so providing support on general data management. We've had a lot of training and provide resources for creating data management plans. And then we have a lot of expertise now in clinical data management, specifically using REDCap. And so we've trained people in data management courses, and we've also we have this REDCap Africa monthly meeting. When it comes to the tools, we know that for genomics, many tools exist around the world, and we really don't want to reinvent the wheel because they've been developed and benchmarked by the best. So what we aim to do is think about, well, which of these tools are best suited for our data, and where there are gaps is to think about either improving on existing tools or creating new tools. So this is just a list of some of the tools that we have been working on in AfSHG. So they cover things from analysis of whole genome sequence data to array data to pathogen genomics data, and then also data interpretation and visualization. And again, we have a training component in here. So we run hackathons to develop these tools so that we have an interdisciplinary environment for our developers to work alongside experts and alongside the biological users. And then we also run analysis workshops for people to be able to run these tools to analyze their own data. So just looking at how we try to improve the tools for African genomics. If we look at NGS data, the steps are align to reference, call the variants, and do some variant annotation. So what we're trying to do is think about how we can improve the reference and how we can identify the best variant calling tools, and I'll talk a bit about those two in detail. When it comes to arrays, obviously there's a choice of array. We've developed the H2Africa array, which is better suited to African populations, but we're also working on what is the best imputation tool, what is the best reference panel. And then for variant prioritization, it really requires access to African data as well as access to global data. So we're trying to provide all this data in a much easier to access format. So starting with the reference genome. So we know that the current linear reference genome is a bit biased and not diverse enough to cover particularly African populations. And we know that the extent of genetic diversity in Africa is extensive. And so the reference genomes are really not complete and not good, do not have good African representation. So in a study by Sherman et al., when they looked at the pangenome, and this was on African American genomes, they found that 10% of the African genome was missing from the reference, and there are many important protein coding genes in this. So in AfSHG, we've got this pan-genome project where we're trying to do two things. Number one is to build a pan-genome like they did in the Sherman paper, but with as many sequences as possible from the African continent. And for that, we're building and then we're building reference graphs. And for these, we're generating long read sequence data so we can get high quality long reads to be able to resolve regions that are typically quite difficult to resolve. So we're generating a pan-genome from these, and we will then look at assessment of variant calling off the pan-genome and the reference graph to see whether they actually do improve the reference alignment and variant calling in African genomes. So we've just recently started working with the International Human Pangenome Reference Consortium. We've started joining their working groups because they are building this new diverse pangenome and reference graph for human genomes. And so it makes sense for us to align our efforts to them. So just to illustrate why we need to do build these reference graphs and think about pan-genome. So this is an example from a bacterial genome, Mycobacterium tuberculosis, that we've been working on. So this is where we first explored reference graphs. And the idea of the reference graph is that you have an alignment. If you see an A, we have an alignment of several different sort of major lineages or major strains. And then you'll see that within that alignment, there are regions that are completely conserved. And then there are regions of variation from that alignment. So in C, you'll see that where those regions are conserved. In D, you can collapse that down to a single reference linear sequence. But then where there are positions that are diverges or major structural rearrangements, you can represent those as a graph and a bifurcation on the graph. So it's a variation on what is the norm. So you have some parts that are linear, and then these pieces that or graph structures that branch out. So for TB, we used these genome graphs and built a pan-genome to do some alignments for a set of transcriptome sequences that we had, so RNA-seq data. So we used the main reference, H37RV, and then some of the clinical isolates and the Beijing strains, because the strains that we were working with in this particular experiment were Beijing. So when we aligned the RNA-seq data to the H37RV genome, we found that actually there was not 100%, I mean, it's never 100% alignment of your reads, but it wasn't as good as it could be. And the H37R genome is actually quite a bit smaller than the Beijing. And when we looked and now aligned to our reference graph, we found that actually that the 4 genes that are all in the operon that happen to be the ones that were important for this particular experiment, because they were significantly differentially expressed during all conditions, happen to be part of this operon, which in H37RV is in fact truncated. It's missing the half of the third and the whole of the 4th gene. So if we had aligned it only to H37RV, we would have completely missed this significant finding. And so when we have mapped it to the full genome, we get much better alignment. So it just shows you that if you don't use the correct reference, you're going to miss data. What if those disease-causing variants are in fact on pieces of the African genome that are not in our reference genome? The other thing we explored was Varian Calling. So there are many different Varian Calling tools that are available, and there are some overlaps, but they are. This we use simulated data. So the golden VCF is what the SNPs should be. But you can see that there are a lot of SNPs that are uniquely called by specific tools. And when we simulated African and European data, high coverage and low coverage, you can see that for the true positives, there was quite a big difference for the different tools in how many variants or the percentage of true positives for European versus African. Percentage false positives also vary by tool and by population ancestry, and same with false negatives. So this is, I mean, and Emil Chimusa talked a bit about benchmarking tools yesterday, and he's been involved in this project as well. There are definitely biases in the tools for the different populations. So another consideration is joint calling. If you do single sample variant calling versus joint calling, you also need to think about variations in the results. So for the genotyping arrays, we're also assessing the imputation. We're looking at the tools for general imputation and HLA imputation. We're looking at different reference panels. What is the impact of size and diversity of the reference panel, and the impact of sample size in terms of the number of samples you're imputing at once. So this is the project, a collaborative project with this group at WITS. And the issue, of course, here is that many of these reference panels are not publicly available. Even the H-Africa one is not publicly available. So they get run as a service, and they're usually tightly linked to particular tools. So it's really quite difficult to assess them side by side. But we've nearly finished our assessment, and we're going to be submitting a paper hopefully soon on that. And then improving downstream interpretation. There are many tools that we're trying to work on that can improve this. But when you look at variant prioritization, this often relies on math data. So you need access to frequency data for African populations at a level of granularity that is not just African. We need a level of granularity that shows you the different regional variations in Africa. This is very limited in terms of its accessibility. If it is available, it's not necessarily accessible, and the granularity, for example, in NOMAD, is not fine enough. So we've worked on the African Genome Variation Database. This is, and it's going to be released soon. So this provides an interface for browsing African data, but with greater granularity. So what we've done is for now, we've pulled in all just the publicly available African data. And we've tried to sort of map this specifically to geographical regions. So it provides for each variant a list of information. So, links to curated databases, information on the frequencies. You can filter the data, so you can find all data from a particular country for a particular genomic region, for example, and then get the population frequencies now distributed now with specific geographical locations within Africa. And then there's also a link to add, to view the nomad data on top of that, which is, of course, much less fine-grained. We also have a map view where you can look at, for a particular frequency, for a particular SNP, what is the frequency across different populations. So we hope this will help to increase accessibility of African frequency data. And we'll also have different levels of access so that the data that can't be made fully public can go in here, but under registered or controlled access. So we're hoping to leverage these tools and the data that we can collect in these tools for, as an example, in application in rare diseases. So we know that for rare diseases, the diagnosis now often relies on doing whole genome sequencing, but you need now good reference genomes for healthy individuals to be able to compare against, to be able to filter your variants and think about what variants might be associated with this disease. So AGBD forms a reasonable option for this. So if you think about the workflow, you'll do whole genome sequencing, and then you want to do filtering of SNPs for the maps and clinical significance, and then the variant annotation. So if we have what we're working on is a tool in AGBD such that filtering can be done, obviously different levels of access. And then also for this, would obviously need to split into cases and controls, because for this filtering, you'd only want to be using control data. And then we're also developing a genomic medicine portal, which is pulling out all that vast amount of information that's out there about what is the clinical significance of variants and putting those into a curated portal. And then, of course, you can now access things like matchmaker exchange. So this is for looking at diseases, particularly rare diseases, if you want to compare your case to something that's been seen around the world. And then you can also run a beacon query, which is we also have available from AGBD. So what is a beacon? So a beacon, if we think about the way people share data, you ideally would go get a data set, download it, and then find your variant of interest. What a beacon allows you to do is a very simple query across multiple data sets around the world to say, for this particular position on this particular chromosome, for a particular genome build, have you seen an A? So you just put in a particular allele. The beacons then go around and search all the data sets around the world and come back and say, on the picture on the right here, you say for these data sets, 1,000 genomes, etc., the result is either yes or no. And so you're very simply saying, you're seeing what is out there without, your data being given away. And there are further extensions that have been developed for this already now, Beacon version 2, where this now includes, you can add phenotypes. You can say, yes, I've seen this, and that was associated with a particular phenotype in my cases, or I've seen this in this data set at a particular frequency. So EGA offers a beacon. If you submit your data to EGA, you can also select whether you're happy to have your data available as a beacon, and this can be under controlled, registered, or open access. So beacons allow you to access very small pieces of information, because sometimes you really actually only want to check a couple of variants that you maybe have got from your GWAS. But what if you want to access larger data sets to do big meta-analyses and to increase your power? So this is just to illustrate why you'd want to do this. We've got a UK biobank data. We did just a quick study of a particular trait in African samples, African ancestry samples, only about 3,500. European ancestry samples, 470,000. So you can see the orders of magnitude of p-values that you get that are different because of the sample size. So you can see the power, how the power changes by increasing the order of magnitude of the sample size. So how do you get to these bigger samples? What does it take to scale? So Ambrose has published this amazing paper on a fantastic vision for sequencing 3 million genomes across Africa, but this is not coming cheap. So they estimate $450 million per year for this to actually happen. And I think this is something that is amazing and needs to be done in African populations because of the amazing diversity we have. So in the Nature paper that was published at the end of last year for the AfSHG Consortium, it showed about 3.5 million novel variants. But even just between the the second last and last population, as you add new population, there were already an additional 6,000 novel variants. So if you try and extrapolate that to over 2,000 ethno-linguistic groups across the continent. As we add these new groups, we're looking at a potential for more than 12 million completely novel SNPs. And so there's amazing potential for such a project to really improve our knowledge on SNPs in African populations and worldwide populations. But if you look at the data itself, the data requirements, so if we, Gerrit has done some calculations. So if you do, 30x coverage, you've got your FASTQs, you use CRAM format, which is compressed BAM, and your GBCFs, for 3 million, you're looking at about 600 petabytes of storage, a bit less if you don't keep the FASTQs, and 105,000 CPU years. So this is basically calculated based on our current reference panel, which is about 5,000, and then extrapolated. When you get to the next step of Genomics DB import, you're getting to, it's a small amount, 36 petabytes and 1,000 CPU years. Then you've got to do the joint calling and another 2,800 CPU years. So you're looking at about 1,500 petabytes of storage, less if you don't have the FASTQs, more than 100,000 CPU years. If we look at the average costing based on the quote we'd received from one of the cloud providers, you're looking at $34 million for storage, but more than $3 billion for compute. So it's a huge computational cost. But these, of course, can be cut down if you do sort of a bit more parallelization, you remove files that you don't need, and you manage your uploads and downloads, because it's our downloads of data from the cloud that are the most expensive. And then you can consider other options like commercial options like Dragon and Sention and Parabricks as different compute processing options. But either way, it needs very good planning, a lot of computing infrastructure, a lot of money, and none of this actually factors in the human costs required in terms of setting up this infrastructure and obviously managing and analyzing this data. So it's definitely something we should take on, but we need to sort of do some careful planning. So what can we do in the meantime if we don't have this huge data set? We can think about using existing multiple data sets. And we do know that there is African data around there, but it's all sitting in silos. And so you want to pull those together to be able to make inferences. So we know that there's a massive power in sharing data sets, but this requires findability of data sets. We have the H-Africa catalog, which shows you how to find the H-Africa data sets, and the International 100,000 Cohorts has built an atlas to find additional cohorts that are existing around the world. You need harmonization of the data. So in H-Africa, we're doing a lot of data harmonization. So we're building standards. We've built standard CRFs with the H-Africa Phenotype Harmonization Working Group. These are all mapped ontologies to make the data interoperable. And we're working on algorithms for actual harmonization of the data. There are additional metadata harmonization efforts around the world. So I'm involved in the Seneca project, which is looking at cohorts across 3 continents. And there we've built a minimum metadata model. And from that, they've actually developed Genomic Cohorts Knowledge Ontology, which maps out the metadata for cohorts. And this has been used to actually populate the IHCC Cohorts Atlas. And we've mapped the H-Africa metadata to Gecko. But there are also other projects like European Beyond 1 Million Genomes, like a big continent that now wants to share data across borders where it can't potentially move. So the other thing about using different data sets is what are you allowed to use the data for? And GF4GH has developed the data use ontology, and we've mapped the data sets to that. So what this enables you to do is to say, this is automatically to be able to query data for what you're allowed to use it for. So for example, find all data sets that I can use to study cancer, and I'm a commercial entity. And so we have the over 200,000 data sets around the world that have been annotated, and some of the H3 Africa data sets too. So the next step is thinking about federated data analysis. And now you've got these, you know what you can use it for. The data are sort of harmonized, but how do you really access and analyze data? And in many cases, and even with the 3 Million Genomes Project, is it going to be that these data sets actually need to stay within their national borders, even though they can be shared? So you need to have a scenario where you can visit the data rather than download and take the data. So Seneca's tried to look at this by building standards for discovering the data in the first place, authorizing particular people to use those data sets through these access committees, harmonizing the data to make sure once you have access them, you can actually compare the data, building the tools to do the analysis, and then having this all designed within a suitable LC framework. And of course, GF4GH standards are used for that. So how does this practically work for researchers? So researchers got their own data set, but there are many other data sets sitting in, behind walls across the globe. So and these are indexed in a catalog that's geo-mapped. So the user will find the data sets that they want. They'll then request access to the suitable access request, the DAC. They'll get access. They'll be authorized. They send their tools to each of these pieces, all these data sets sitting around the world. And then all that comes back is the results. So no data has to move, just the results, and then they can do the analysis. You can also tools now to be able to extract pieces of the data. So if you just want chromosome 6, for example, and ideally you'd want actually these data sets to have a public component where summary data already, and then you can just access that. So we're thinking about this within a single framework in Africa. So, the idea is that you have all these data sets, if that scenario before was when they're sitting in different places, but what if you could have them all in a single framework? And then you have a user workbench. The user comes in, they log in, they find the data sets they want, they get their access, they find the tools they want, they send the tools to the data, and they run everything on the cloud computing. So this infrastructure actually exists. So Terra is at the Broad Institute, implements this kind of system, and it's been used for high-profile projects like All of Us. So we think this is a good model for African data, and we propose it actually for the NIH DSI Africa Open Data Science platform. So once you have the data, obviously the data are bigger and more complex. You've got all sorts of different kinds of data that you can link to. And so we need to start thinking about implementing these real data science tools. So machine learning is not new to bioinformatics. There are many different applications all across bioinformatics and how machine learning has been used. And if you look at genomics, it's a particularly good use case because you've got hundreds of cell types, 3 billion possible positions for thousands of participants, hundreds of biochemical measurements that can be made. And you can use deep learning to understand these, to build predictors, to identify potential drug targets. So you can use these to do diagnosis and accelerate drug discovery. So we've got an ML project in H3 Bionet where we're trying to build tools and glossaries and provide support and actually implement tools to see what are the best sort of ML tools for the best data sets. So we have these a glossary, we're building training materials. We're trying to find, we've actually got some data sets that are now being applied. So we have building the expertise for real data science. So just to finish off then, because I know my time is up, genomics activities in Africa are increasing, not just with human genomes, but also pathogen genomes. We've seen a huge acceleration in the sequencing of the SARS-CoV-2, particularly by Christian Happy's group and Tulio de Oliveira in South Africa. The Africa Biogenome Project's being launched to sequence all sorts of diverse flora and fauna in Africa. So we need to make sure we can scale, that's infrastructure and skills to keep the data and analysis on the continent. We want to be able to develop platforms for responsible data sharing, embrace data science technologies so we can really do downstream interpretation and integration of data. And I think Africa has the necessary foundations. I think through what we've built over the last 9 years, we really have a solid base to be able to embrace this. And then the new initiatives, obviously, that are going to really push this is the NIH DSI Africa. So with that, I'd like to acknowledge the H3 Africa and H3 Bionet Consortium and colleagues on the Seneca project. And thank you for your attention.
data science in Africa. And it's always amazing to see the way you can conceptualize things and show it on a very simple way, because it's actually very complex data. We have for now one question, someone you know well, Gaston Mazandu. Great, dear Professor Nikki, for the great presentation. What is the biggest impediment for data science and genomics in Africa today? So that's the first question. Is there a limited amount of existing data and data sharing process still to be refined, or perhaps flexibility of PIs? Another question, he's very prolific this morning. What may be the way to overcome this impediment?
Okay, so yes, so I think the biggest challenges, so we do have internet challenges, so basic computational challenges. If we're going to have to embrace cloud technology, I think that's inevitable. There is some resistance to doing that because of the concept, the perception that this is now sort of opening your data up. We have national laws that might not allow data to move across borders, and therefore, we'd have to have local cloud providers. So I think the computational challenges are huge, but the other challenge you mentioned, I think, is maybe one of the bigger challenges, is actually getting the sharing of data, responsible sharing of data working, so that people realize the value of sharing their data and having the facilities to be able to do that. So I think, and we've built the skills. So I think we have the foundation to get this going. It's a matter of the will now to move with the times and to embrace these new technologies and use the amazing, valuable data that is already being generated as maybe use cases to show how data sharing can actually push knowledge on the continent.
So there is no further question, but I have one question for you regarding sharing of data and open data science in general. Equity question. I think that there have been some resistance, specifically amongst African scientists, and specifically within the consortium we know very well, which is the H3 Africa. And we still have some resistance of getting the sample and the data to the biorepository and to the how do you think we should go about equity? Who actually benefit of the open data science? And what are the ways to address equity in open data science?
So that is a very, very important question and a very difficult question. We find, when we, even just within the consortium, when we're trying to share data, we say, okay, we'll do it through a capacity building exercise. And so things go much slower, but I think we have to have that patience and invest the time to make sure that the people who are the data owners have the skills and the ability to analyze the data and be involved in these collaborations. So, even if when people come in and want to use the data, embrace, include the people who generated the data, let them get the value out of it as well, let them learn so that next time they can analyze their data and also exploit the data. So I think having, capacity building components for the sharing of data, but also think about the endpoint, what is going to benefit the original participants the most? If you hold on to that data, you don't have the skills to turn it into something, a health benefit for your original participants, then that's also not fair. Mm-hmm. So think about who you can partner with to derive the best benefit out of that data in terms of increasing knowledge, but also getting a benefit back to those participants. And, this capacity building, partnering, equal partnering, I think is the way to go about it. And transparency, open transparency, about what the intentions are with the use of the data.
Okay, thank you very much, Nikki. I think I'll go back to the discussion forum again to see if there is any other question. There is no more question. We are 7 minutes about the prescribed time. We start a little bit late. So thanks, Nikki, for this excellent presentation. We will now take the opportunity to introduce Professor Dave Valley, that he will speak, give a few perspectives of genomic training, and specifically with regard to the Bahab or short course in the US. For those that are not aware, this is a course that have been there for the past 62 years at least, initially developed by Professor McCrissick, that we consider as the father of modern medical genetics. It is considered by many geneticists across the world as a rite of passage for geneticists. And the course actually have met some mitosis, and the European course that's been running in Bertinoro in Italy for the past 30 years. Is a derivative of that course. And the invitation of Professor Valley here is purposely around the wheel of the African Society of Human Genetics to develop a similar thing with the help of the original course from Bar Harbor. Dr. Charles, who is the Vice President for Education for Jackson Lab, Professor that I don't think he'll be there today, but Professor Valley is the Director of Genetic Medicine at Johns Hopkins University and the Director of the Institute, McCusick-Nathan Institute for Genetic Medicine and Johns Hopkins University. And he will give us some clue on how the next generation of training for genetic medicine and human genetics will look like, specifically for the benefits for us African scientists. Thank you very much. Prof. Valley.
Good morning, Ambroise, and good morning to all of you. I hope everyone can hear me. I do not have slides, but I will take this opportunity to talk a little bit about the challenge and value of education and how necessary it is to move our field forward. Dr. Mulder has given us a wonderful talk that highlights both the opportunities, the tremendous opportunities, and the challenges of acquiring genomic information across the globe, and specifically across Africa, so that scientists as well as scientists elsewhere can compare their data and use this data to advance our knowledge about genetics and biology and disease. Of course, in order to do this, we have to have a large workforce of students and trainees that know how to use this information. And the value of education, if you think about it in that way, becomes clear. The other challenge, of course, is to take advantage of the full opportunities afforded by genomics in medicine, is to educate not only geneticists and future geneticists, but also to educate our colleagues in medicine, so that physicians doing internal medicine or infectious disease or orthopedics or general surgery or whatever incorporate genetics and understand human genetic variation and how it applies to their particular areas of medicine. So we really have a need for enormous educational activities to harvest the value of the information that the rapid technological advances allow us to accrue and to analyze. So it's a really exciting time for education. I'll just say a word about what we're doing here at Hopkins, and I'll talk in some detail about what Ambrose mentioned, what we call the short course or the Bar Harbor course. Bar Harbor is a small town in northern Maine where we've been teaching genetics for many years. But at Hopkins, this challenge involves bringing students into our field and then bringing them up to speed so that they can apply their creativity and their knowledge to questions related to genetics. And in addition to the students, it requires reaching out to our colleagues, many of whom have had some training in genetics when they went through medical school, but that training is, of course, immediately out of date because our field is moving so rapidly forward. And so we do need to have a robust interaction with our colleagues across medicine to help them realize how they can use this information and how they can use it to improve their ability to care for their patients. So that's an ongoing challenge. It's very interesting, and it means that genetics spreads across all of medicine. I don't need to tell the folks in this audience that, but I do need to remind my colleagues here at Hopkins of that feature all the time. So as Ambrose said, the idea to have a short course in that allowed a very intense experience in genetics to bring people up to speed, to allow them to understand and to use these tools, began actually in 1959 And I think the major credit goes, as Ambroise said, to Dr. Victor McKusick. So Victor was born in the state of Maine, which is in the top northeast corner of the United States. And it's an area that is heavily forested and has sparsely populated. And of all things, Dr. McKusick was born in a very small town, just maybe 3 or 4 houses, in the central northern Maine, a place called Parkman, Maine. And he was one of identical twins. And Victor and his brother Vincent grew up in this very small town. They went to what we call a one-room schoolhouse, where all the kids in the region were taught in the same classroom. So the pictures I've seen, the class was usually somewhere between 10 and 20 kids. And I just imagine what it was like to teach that group, having Victor McKusick and his identical twin brother, Vincent McKusick, who both obviously were very smart and I'm sure challenged their teacher all the time. Vincent went on to become a distinguished judge. Victor got, as he said, bit by the medical bug, and ultimately he made his way to Johns Hopkins School of Medicine in the very early 1940s, and went through Hopkins Medical School and residency in internal medicine, and was going to do a career in cardiology. However, he stumbled, or he picked up a case of Peutz-Jeghers syndrome. I'm sure many of you know that. It's some abnormal pigmentation and an increased incidence of GI polyps and so forth. And he became fascinated in that family, and he interacted with Dr. Peutz, and together they published a very important paper in the New England Journal of Medicine on the extended family that Victor identified. And that switched his attention from cardiology to genetics. And just at that time at Hopkins, one of the professors who ran a chronic disease clinic was retiring, and the chairman of the Department of Medicine asked Victor to take over that person's clinic. And Victor said, I will do that, but I want to switch it from the chronic diseases that my predecessor was dealing with, which is largely syphilis, actually, to genetics. And the chairman of the Department of Medicine, A. McGee Harvey, said, okay, and which is pretty amazing because there was no other clinic in the United States that was focused on genetics and medicine. So Victor started that clinic in 1959, or 1957, I'm sorry. And one of the first things he did is to try to get money from NIH to support clinical research in that clinic. And his operation was visited by a committee from NIH to evaluate the program. And a member of the committee was a man by the name of Earl Green, who was the director of the Jackson Labs, which was a mouse genetics lab in Maine, of all places, on the island of Mount Desert, which is just off the coast of Maine, in the town of Bar Harbor. So that's how the whole thing got to be located at Bar Harbor. Green saw the common interest between him. He was interested in mouse genetics, and McKusick, who was interested in human genetics. And he also noted that McKusick was from Maine. So he said to Dr. McKusick, why don't you come and visit us next summer? And so that led to Victor and his wife, Ann McKusick, also a physician, visiting Bar Harbor in 1959. And they had a meeting with the director of education at the Jackson Labs. And together at that meeting, they decided to develop a course that would teach genetics to academic physicians, actually, to sort of magnify as rapidly as possible the number of individuals who knew something about genetics as applied to medicine, and allow these people to go back to their respective institutions and teach genetics to their faculty and their colleagues. So the original goal was to provide the students with the up-to-date knowledge in genetics, and to give them the tools that would be required for them to become effective teachers of genetics at their own institutions. The first course occurred in July of 1960, as Ambroise said, 62 years ago, and we just completed the 62nd course in the end of this July. Over those 62 years, the course has consistently made an effort not only to teach the teachers, but also to make everyone aware of the very most up-to-date advances in genetics. And so if you look at the curriculum of the course and the content of the course over those 62 years, you can see that it's really the history of human genetics over that time frame. Initially, the course was divided in alternative years into a mouse course and a human course, but within a few years, it was recognized that many of the advances in either mouse or human genetics were very relevant to the other field, to the other group. And so within 10 years or so, then the whole thing was amalgamated, and we just mixed human and mouse genetics as used by the speakers for the course. And so we no longer separate it into mouse and human genetics. The content of the course is organized. Right now we have a committee of organizers. I've been a co-organizer since I've been an attendee since 1976 and a co-organizer since 1990. And the general scheme, I won't go into it in any detail, but the general scheme is the first week is more about the basic science of genetics and genomics, including many of the computational technologies that Dr. Mulder mentioned and the population genetics aspects of that. And then in the second week, the course merges into a more clinical genetics course, focusing how this information can help us understand human genetic disease and the susceptibility to various genetic phenotypes and complex traits, the genetic component of that susceptibility, and how the genetic component integrates with the environmental exposures to determine whether people either remain healthy or get sick, and how we can keep people healthy by by dealing with their increased susceptibility to try to keep them healthy. It's a pretty intense course. The past course had about 80 lectures distributed over the 2 weeks, so it's quite intense. And up until 2 years ago, up until the pandemic, of course, the course was in person, primarily at the Jackson Laboratories, which is a very modern, world-class, in fact, arguably the most, one of the top 1 or 2 mouse genetics labs in the world. So it's a very modern facility, and it's on a campus, a relaxed campus, so it's very comfortable in a beautiful place along the coast of Maine. So you're on the seaside, but you're also in the spruce forest. And so people spend a lot of time listening to, as I say, intense lectures, but there's also time to stroll about and to get to know your colleagues from all over the world. When the course was entirely in person, the audience was usually around 110 to 120 students, and the students were quite heterogeneous. They included pre-doctoral students, postdoctoral students, junior faculty members from various parts of the world in academia, and usually some individuals from the private sector looking to apply genetic knowledge to some aspect of their business. So it's quite a heterogeneous mixture of students. And as I said, about 110 to 120. There's a lot of, in addition to the lectures, there's a lot of discussion time, group discussions, as well as individual one-on-one discussions. The faculty make themselves very available to talk to students one-on-one over lunch or over coffee break or for a beer in the evening, all different kinds of things. So it's really a very potent learning experience. In the recent years, we've been very eager to disseminate this learning experience to a broader audience. And as Ambrose said, one example of this some years ago was led by Giovanni Romeo, who developed a so-called European short course. Giovanni trained at Hopkins, early in his career and knew of the short course and worked with Victor to start the Bertonoro course in Italy, I guess about 30 years ago. And so that continues to move forward. But we realized that as good as these in-person courses were, we could only reach a relatively small number of individuals. Over the 62 years of the short course, we've had a little over 8,000 students total. So we've been exploring, as the internet capabilities came online, we've been exploring the use of the internet to disseminate the course. And of course, the pandemic has really pushed us. And last year, the course was virtual for the first time, all virtual. And we used roughly the same program and the same speakers, but we took advantage of the same kinds of informatics that this meeting is being broadcast with to make it accessible to students from all over the world. And last year, instead of our usual 120 students, we had more than 1,000 students. And they came from 6 continents across the world. And we did a pretty good job. The lectures are actually fairly straightforward, but of course, the question and answer and the discussion sessions and so forth are a little bit more cumbersome. But we did, I think, a pretty good job of learning from that course that year. And this year, again, we were all virtual. We had this year, I think, a little bit more than 500 students, and again, from 6 continents. And I think we did a better job at facilitating the discussion groups. So they were in general quite lively, and people got a chance to ask their questions either in person or through email and so forth. And we plan next year to have we hope to have a more hybrid course of in-person and virtual. A few years ago, I think now about 4 years ago, we began to explore the idea of having a much greater interaction with geneticists in Africa. And this idea came through our interaction between Professor Charles Ray and myself with Ambrose and his colleagues in Africa. And we met, I think, I don't know, 3 or 4 years ago at American Society of Human Genetics meeting and discussed this possibility. And we thought that we would start off by Ambrose would send some students from Africa, and we would begin to incorporate African geneticists in the faculty of the course, and then expand this sort of foothold each year. This year, we had a 2-hour work session one morning on genetics, human genetics in Africa, and I think we had 5 or 6 speakers, including Amboise and several other colleagues from Africa. And that was a very exciting session. The feedback, the course is evaluated by an outside agency, and the feedback that we've gotten from that evaluation was that it was extremely successful. We do, a segment of the course has traditionally focused on population genetics and the extent of variation around the world. So having a much more prominent activity in African genetics, where there is, as Dr. Mulder emphasized, so much variation in human genetics, was really fit into the course very well. And of course, we also have a large segment of the course that deals with rare diseases, and the speakers at the African part of the course emphasized their work on identifying and rare genetic diseases in Africa, and advancing our knowledge in those disorders and learning how to treat them and keep those individuals healthy as best possible. So, and going forward, we plan to continue this activity in the virtual course, but at the same time, we plan to hope to develop an on-site course in Africa that will share some of the professors that are experienced from the Bar Harbor course, as well as the colleagues in Africa who know obviously much more about African genetics and genomics, and the kinds of phenotypes that are found in Africa and how they relate to what are found in other parts of the world. So we envision going forward easy access to students in Africa to the sort of parent Bar Harbor course, as well as developing a course in Africa. The details of that are still under discussion, but I think it's fair to say that we envision that it will perhaps move around Africa. It won't be in one place each year, but move around to different parts of Africa so that we can reach out to as many students as possible. And it will provide the one thing that the virtual course can't do, which is the opportunity for in-person interactions, which turn out to be a really valuable component of the course, allowing colleagues to get to know one another and to think about joint projects and how their research and their clinical activities can synergize one with another. So we're very excited about this. We're committed to this. We just had the short course organizers meet every 2 weeks to plan next year's course, and we had a meeting yesterday morning, I can tell you. And a good part of that meeting yesterday morning was to discuss how we were going to continue our expansion of the interaction and the synergism with African genetics going forward next year. So we're very excited about this. Lots going to happen, and we hope way and disseminating our knowledge of human genetics through the short course in this way will spread the word about genetics and knowledge about genetics across the world, and particularly to Africa. And we'll just be following in Victor McCusick's original goal, which was to teach the teachers and give you folks in Africa the chance to benefit from this course, and then to turn around and teach your own students based on what you learn. So that's a very high-level overview. I'd be glad to answer any kinds of questions, and also very eager to get any suggestions or input so that we can make this the very best course possible. Thank you very much for the opportunity to speak.
Yeah, thank you very much, Dave, for this excellent overview of what have been the development on the short course and McCusick course, and what would be the perspective in collaboration with Africa. I recognize in the room Professor Gada Elkama from Cairo, that have been in the forefront of the conversation on developing this course, as we plan to have the first in-person meeting, probably course in Cape Town, and the following one in Cairo. And then we will move around as we go. In the planning of this course, I will cite also Gida from Mali, and the intervention of other colleagues like Sonia Sonia from Tunisia, and Christian Happy from Nigeria, Neil Hanshat that actually presented at the platform from the United States, originally from Jamaica. So there is already some movement, and also a few colleagues that have participated, and hopefully will form a nucleus around it. Is there any question? Please put the question in the chat. I should have said that before. Okay, maybe the first question to you, Dave, when will you, let's say if COVID is kind to us, will we be ready to start a first in-person course on this place on the continent next year or the following year, based on the planning that we have now?
Well, actually we discussed that yesterday, and we need to get the planning group for the African course together. Obviously, much depends on the pandemic and the ease with which one can travel. We would like to do it sooner rather than later. I don't know if we think, it sort of depends on what's feasible for next year. I'm sort of guessing, I don't know what you think, Ambrose, I'm sort of guessing that we're maybe 1 year away from it.
Yeah, I would think 1 year away, getting the situation to be more stable. Gada, what do you think? I can see your words in the chat around the presentation. Congratulatory. Are you ready for 1 year away from now?
By 1 year, I mean not this coming summer, but the following summer.
The following one.
Yeah.
I think there is no other comment. If there is no further question, thanks. Oh, okay, I have two questions here. Julie is suggesting to link the course to, I think there is two acronyms that I can identify, AfSHG, Education Committee. Probably I'll ask you about that later, Julie. There is a question from Dr. Mazandu. I know it's a question from the previous presenter. So if there is no further comment for now, we will move to the next presentation because we are 3 minutes above the time. Thanks a lot, Dave. Thanks really to be present to this, and being here out of your time really means a lot to us. And we hope that we will move forward as quick as we can to have another equivalent of a European course from Bar Harbor to Africa.
We're very eager to see it happen.
Okay, thanks a lot. Our next presenter is a very different type of presentation. I will warn you that it's a promotional presentation. It's our industry partner, and this year 54 Genes. That is one of the biotech. Yes, okay. That is one of the new biotech trying to take advantage of human genomic variation on the continent to translate it into research and development and commercial product. I think 54 Gene have had a lot of traction, a lot of publicity across major outlets like Nature, and they have been supporting this meeting this year, and we hope to have them as a partner for the next years. And this is an opportunity for 54 Gene to tell us more on what they do and what they envisage to do in terms of industry and genomics in Africa. To represent 54 Gene, we have three speakers: Dr. Aminu Yakubu, Dr. Kom Odushlen, sorry for the pronunciation, and Dr. Jumi Popula. I think the floor is yours.
All right. Hello. So we're currently waiting for our Chief Operating Officer Delani to get access. We're both struggling with that. Aminu, I believe you're going to share some slides.
Yes. So the slides I think are on. Slideshow. Okay. I've done that now.
Great. I mean, Aminu, do you want to give a few words just introducing the company?
The slide showing though. Just wanted to be sure.
Yes,
okay.
Thank you.
Thank you. Yeah. So unfortunately, I think Delali is unable to join because of connection challenges. But I think.
Yeah, I think you can go ahead and say a few words.
Okay. Thank you very much, and thank you, Professor Wonkum, for that introduction. We are happy to be able to make a presentation during this session and actually to be members of the family of human genetics in Africa. Today, earlier, 4 of us are supposed to be on this presentation, but right now only 3 of us have been able to join, and the challenge has been due to internet connectivity. Be that as it may, I think we will just start with the introduction of the three speakers that are here. And I'll start with myself. I am Aminu Yakubu. At 54Gene, I lead the research, governance, and ethics team. And largely our focus on that team is to ensure proper governance of our research operations, our biobanking operations, and our sample and data sharing policies and practices as well, while ensuring that our research studies are done based on highest ethical standards, both locally and internationally, understanding that there are different regulations in different countries. So our effort is also making sure that we work in line with those country-specific policies. Prior to 54Gene, I had worked with the US Centers for Disease Control as a science program specialist, largely making sure that publication products going out of the US CDC in Nigeria meet the highest standards of the US CDC. And before that, I worked with the National Health Research Ethics Committee in Nigeria for about 12 years, ensuring ethical practice across all the ethics committees in Nigeria. Colum?
Yeah, so my name is Colum Odulana. I head up the Genomics and Data Science group at 54Gene. And our role is to sit and work very closely with Jumi and her team to process, sequence, and array genotype data that comes through, and to perform analytics in the cloud, bring that through to GWAS at the end. Jumi.
Thanks, Com. Hello, everyone. It's a pleasure to be here. My name is Jumi Coppola. I head the molecular genetics and biobank operations at 54Gene, and we're largely responsible for setting up of the infrastructure for the high throughput genetics laboratory in Nigeria, as well as expanding that to other parts of Africa. We've also established a biorepository of multiple sample types, and again, that has been driving our research efforts successfully in the last couple of years. And prior to joining 54Gene, I was part of a collaboration with the Cancer Research UK, the Institute of Cancer Research, and Merck Pharmaceuticals in Germany, looking for best practices and approaches to drive drug discovery successfully from bench to bedside. I'll hand over back to Aminu to go through the overview of the company.
Thank you very much, Jumi. I was waiting for the mic to come on. As some of you may already know, 54Gene is a health technology company enabling precision medicine in Africa. And we are very focused on improving precision medicine. And we're trying to achieve that by building research cohorts that unlock scientific discoveries, while also ensuring diagnostics and treatment outcomes are improved in Africa, and of course the global community. And what we're doing now is really investing in genomic infrastructure and collaborating with ecosystem partners to build sustainable solutions that are beneficial to patients and the scientific community. We have a state-of-the-art molecular laboratory here in Nigeria, which is the first we have established, that has capacity for high throughput sequencing and genotyping, and of course also other molecular diagnostics capacities. Dr. Jumi will say a little bit more on that. What we have also done is to establish a set of capabilities and infrastructure that are designed to drive research and healthcare innovation in Africa. So what do we have? We have a biobank, and right now we have one that has a capacity to store about 300,000 samples. And that's just for our operations in Nigeria. We have plans, and the plans are already in advanced stages, to also commence operations in a number of other African countries in the years to come. Right now, there are a few African countries that are already in the pipeline that we've reached advanced discussions with, based on mutual areas of interest. What we also have is a very strong genomics operations department, which Dr. Jumi will talk more about, that has been capacitated with NovaSeq 6000 and iScans, able to conduct WGS and WES targeted sequencing, and also a range of molecular diagnostics, like I mentioned earlier. One other area that we've actually identified the need to create impact is access to molecular diagnostics for especially diseases that are underdiagnosed in a number of African countries due to limited access to such diagnostics, with quick turnaround time in order to enable faster diagnosis and clinical decision making. We also have a clinical program through which we have access to over 100 clinicians that are both working as primary and primary principal investigators and co-investigators across more than 30 tertiary and secondary sites at the moment. We have a strong data science team that is led by Colum, and Colum will tell us a little bit more about his team. What we are doing in that is actually establishing, world-class capabilities for data science, bioinformatics, capable of running analytical tools and visualization of genomic, phenotypic, and clinical data to enable GWAS and FEWAS studies for translational research. And the last, but not the least, is we have built in-house capability with biologists that are able to carry out target validation and develop company and partnership programs. Jumi, so this is, just pictures about some of the things we have in a molecular genetics laboratory. Jumi will throw more light on those. And Column. And there's a capacity building also for molecular genetics training academy that we have, that has been established in 2020, that is training young scientists interested in building a career as molecular geneticist to help create and foster their vision. And we can support researchers in many ways, as it were.
Jumi, we don't hear you, sorry.
Thank you, Aminu. Can you please go back one slide? I would like to give everyone a sneak peek of the facilities and infrastructure in the laboratory. Thank you, on here. So just to expand a bit on what Aminu has explained and introduced earlier, the variety of sample types available for research diversifies the type of data you can generate from a certain population. And to date, our biobank is a biorepository of multiple sample types, ranging from blood to urine to saliva to fixed tissue, and in some cases, fresh tissue, all of which will help us drive meaningful research to understand the drivers of health and disease in the African population. The biobank is a high-capacity biobank, as Aminu has mentioned. It's also mirrored to mitigate against any risks in the event of power or adverse conditions as might happen. And so we have two operational biobanks within Nigeria at the moment. And as Aminu mentioned earlier, we're planning to expand on that in the near future. Apart from that, we have a high capacity laboratory that is able to automate processes for extraction of nucleic acids from the multiple sample types within the biobank, whether that's DNA or RNA for downstream analysis. And all of these analytes also will be quality checked using our state-of-the-art bioanalyzer or capillary electrophoresis-based techniques, again, to ensure that the quality of the secondary sample obtained from our biobank samples is of the highest standards and will give robust and reproducible data in the downstream processes. With regards to the types of applications we're able to offer within the laboratory, we have multiple sequencers from Illumina that are capable of doing whole genome sequencing, whole exome sequencing, as well as targeted sequencing. So we have a NovaSeq 6000. We also have a NextSeq 550DX geared towards the molecular diagnostics, as well as a small benchtop iSeq 100, all of which allows us to have different throughputs to address different projects as they come along. And Com will show you later on some of the progress we've made on the whole genome sequencing front using our high throughput NovaSeq sequencer. In addition to sequencing, we also routinely genotype the samples that we have within our biobank using a combination of commercially available microarray chips from Illumina, but also plans are underway to start to interrogate these samples using our own custom chip. And Com again will expand on that in the next few slides. I have one more slide to just showcase the amount of training and investment in the staff and in scientists within the African continent that we've done. Last year, in the middle of the pandemic, we realized very quickly that we needed to expand on the number of molecular scientists available. One, to help fight the pandemic, but also to drive all the research questions that we had imminent. And so we set up a molecular genetics training academy, which is a combination of virtual and in-person approach to train scientists on the basics of molecular biology and genetics and their applications. So scientists had the opportunity to do the theoretical and practical training. And to date, some of these scientists have gone on to oversee our COVID-19 diagnostics laboratories, of which we have 5 of those within Nigeria. We've trained even non-molecular scientists on the basics, but also we didn't want to neglect the fact that we have very experienced scientists who needed to augment their training on next generation sequencing applications to be able to drive the molecular genetics laboratory and answer some of the research questions we have. And so a combination of virtual training nationally and internationally, as well as in-person training in the United Kingdom for some of our scientists, all ensured that we had well-rounded, highly trained scientists to drive the work that we intend to do. And so with these offerings, we're also able to support African researchers via training and internship opportunities within our academy in Nigeria, and eventually when we set up these academies in the countries that we will collaborate with. In addition, we are always willing to collaborate with principal investigations to answer some of the research questions that we have, especially those that are African-centric, that will allow us to better characterize the African population and bridge that gap that exists in the inequality in the healthcare and even in the amount of genomic data from Africans. And on that note, I will hand over to Dr. Com. Thank you.
Thanks, Jimmy. Right, so I'll give a quick overview of the Genomics and Data Science Group. And as I mentioned before, we sit between the lab and translational genetics and discovery divisions. And what we do really is we've kind of 2 functions. One is bioinformatics, data science, and the second is analytical genomics and kind of gene discovery, target discovery functions. And so a lot of, we're new, we started this year. A lot of our efforts have been in building robust, scalable pipelines, in AWS, and and that are reproducible, scalable, and with good provenance, and we can track everything that's going on And so we've really been focused on building best practices GATK for the whole genome sequence data. And now we're moving towards developing imputation and GWAS analytical pipelines to follow that up. And then the third component is vetting and assessing the quality of the phenotype data that emerges, and just making sure that the inputs to our discovery are pretty clean and sanity checked. Okay, and then next slide, please, Amina. Okay, so one of our earlier efforts this year was to design a pretty low-cost array that could work with the sequence data that we have, and allow us to basically use our sequence data as a imputation reference, coupled with our array to allow us to perform GWAS at scale. And so the idea here was to largely seed the array with, backbone content from diverse populations. So we use the ADPC and mega array backbone content, and then an additional bolus of variants reflected several sources from GKB, GWAS catalog, African-specific EQTLs, aims that we identified from our sequence data, variants of interest, variants that were deleterious in our own sequence data, and then high confidence ClinVar variants. And so the idea here was that the content that we had added was kind of taken directly from 1,000 sequences across for which we had 50 ethno-linguistic groups, and we were able to see identify markers that were stratifying across those. So we think that content will be quite useful going forward, and as we continue to scale up, and as we continue to expand into additional countries, the objective there would be to inform the customer array with content that is specific and unique to those populations. Next slide, please. Okay, and just a quick plug that we have a few open positions. Taking up the kind of engineering division, also specifically focused on rare diseases, and then lastly, kind of classic GWAS and imputation positions as well. Okay, and then just kind of a teaser, and I'll go really quickly through this, but next slide, please, Aminu. Just specifically looking at the sequence data for which we've been looking at for the last few months. In the top left, it's just an example of what we can do. And here we're using cross-population extended haplotype homozygosity to identify variants that are under selection in one or the other population. And here we're using ethno-linguistic groups from the north and the south of Nigeria, and we can see some well-known loci are included here, and I haven't annotated them. It would take a long time to get through them, but so we do see kind of unique stories emerging from these different linguistic groups. On the top right, just showing the principal components plot. And one thing that's very interesting to explore is in these top the more prevalent ethno-linguistic groups in our sequencing data, we do see kind of broad themes emerging and kind of distinct clusters, and that's also emulated in the admixture plot, bar plot that you see just beneath it. And then in the bottom left, we're interested in stories, for example, COVID risk or protective variants and haplotypes that are enriched or not enriched, and just shedding some insight into these quite topical variants, and what are their frequencies across different ethno-linguistic groups in Nigeria and beyond. And then lastly, it's little is really known. This is the bottom right in terms of Neanderthal admixture. Little is really known about the level of Neanderthal or extinct ghost lineages introgression into African populations, and we're very curious into kind of exploring those and seeing that those kind of introgressed segments fix and why they were fixed, and did they confer specific protections historically. So a lot of kind of pop gen going on at the moment, and we'll move to kind of larger scale target discovery over the coming months. So that's it for me. Thank you.
Thank you very much, Colum. And I will be talking about the African Center for Translational Genomics, which is an initiative by 54Gene, in order to largely create impact. So the idea is we are focused on adding value and working together with our collaborators and young scientists in Africa. Hello? Oh, okay. Thank you. So once again, our effort through the African Center for Translational Genomics is really to create sustainable impact. And by that, what we mean is actually working together with young scientists, empowering them so that they can become the future of genomics research in Africa, largely contributing to some of the great work that has been done by other initiatives such as the History Africa. But we also are focused greatly on on building their capacity through PhD scholarships, some internships, and some small grants, for small research studies. And but closely linked to that is our effort to also promote translational research and precision medicine. And with that, we're looking at partnerships, partnership opportunities across different African countries, because we know that there's a lot of thought leaders doing great work out there. And our vision through our translational research and precision medicine is, in addition to our own in-house grown initiatives to power translational research and precision medicine, is also to see how we can work together with other collaborators in translational research. We want to be able to ensure that better efficacy of medicines for Africans and the greater population. And I think the importance of that, looking at what we know right now about some of the drugs that have been developed, cannot be overemphasized. But also through precision medicine, we want to add value care that Africans are receiving by enabling individualized and precise care that utilizes patient-unique genetic identity. What are some of the things that we're doing through that? And I think I may have mentioned that already. We're trying to achieve that through grants to researchers in Africa for primary and secondary research. We are collaborating, doing collaborative trainings, and interested in collaboration to build capacity in statistical genetics, genetic epidemiology, and computational biology. We're also already providing internships, and part of that is what Jumi has also discussed, part of the academy, but internships across all our pipelines to deepen skills in molecular genetic analysis, then also promoting awareness and academic discourse on ethics of genomics research. So what we've realized, and which is not news actually, is a lot of the literature on the ethics of genomics research has been driven mostly by colleagues from the Western countries. What we're trying to see is how we can get Africans to, how we can also improve the voice or increase the voice of Africans in discussing ethical issues in genomics research, as it were, just in addition to some of the initiatives that are out there. Some of the milestones we've achieved already is this year we are awarding 4 PhDs. The call for the PhD grants just closed in end of August. And we have awarded a postdoc fellowship. So our PhD is Pan-African, so it's not limited to just Nigeria, but it was open to all Africans. And then we're also providing writing support grant to discuss LC issues in genomics research. So this year, we are focusing on our writing support grant to discuss benefit sharing. We believe that there's still a lot to be discussed, especially hearing from African perspective of African bioethicists, and other practitioners, and we want to power that. Based on, in terms of some of our collaboration to drive translational research and precision medicine. In Nigeria, we have established the Non-Communicable Diseases Genetic Heritage Study Consortium. This is a consortium between 54 Gene and leading government research institutions in Nigeria. So the Nigerian Institute of Medical Research and the Nigerian National Biotechnology Development Agency. So there is a Center for Genomics Research and Innovation at the Nigerian Biotechnology Development Agency. We are working directly with that Center for Genomic Research and Innovation. And the whole idea through that collaboration is to see how we can together work on high impact genomic research studies that could also, the findings of which could help improve public health practice, in addition to bringing on board new knowledge that could improve healthcare for Africans. We have 2 large studies in that collaboration. One which is hospital-based, which is hospital-based, and through which we are looking at major non-communicable disease markers. So we are looking at cancers, we're looking at endocrine diseases, diabetes and thyroid. We are looking at neurodegenerative diseases, and we're looking at sickle cell diseases as well. We also are expanding that focus starting this year to look at additional non-communicable disease markers, and also to look at rare diseases, which we believe is also an area that is underdiagnosed, and people are not receiving the right care they ought to have received because of essentially that level of poor diagnosis, but also poor awareness of what rare diseases are. And we're looking at also, in combination, being able to provide an environment where people with rare diseases can feel at home and can feel that they are not left alone, that they are really part of the healthcare system, and they are being cared for, just like people with other diseases. Some of the other initiatives we have right now is a collaboration with the Tanzania Human Genetics Organization, which we have just, we are working to see how we can drive genomics research also in Tanzania. There are other 2 more countries that we are in consultation with. And again, our focus is always to work within areas of interest, primarily to the country partners, but of course, areas that we have also shared interest in. And that area simply is areas of priority to Africans, and in health issues that are not receiving, or that have not yet received the adequate attention that they deserve. So this is just a very important, philosophy that we have at 54 Gene, talking about our commitment to our brothers and sisters in Africa. So the most important thing is we want to be able to do what we are doing together with our brothers and sisters in Africa. Just like the saying goes, if you want to go, so there's the African saying that says it's very important for people to work together in order to go very far. And really that's our commitment. We want to be able to go far by working together with scientists and young African researchers, but also working together with the patients or people that are with the healthcare system, as the case is. So looking at 5 core commitments. 1, we want to be able to advance better health outcomes for all Africans through precision medicine and early diagnosis. We want to be able to address the high unmet need for novel therapeutics. We want to be able to increase ethical genomics research conducted within Africa. We want to be able to ensure an African footprint for sustainable growth and job creation. And we want to also build multi-sectoral partnerships for rapid healthcare delivery. And I think all of these points have actually come through some of the bit of things I have said in my presentation, and of course in Columns and Dr. Jumi's presentation. So our collective and collaborative, our impact is collective and collaborative, requiring partnership and participation. And this is just really emphasizing our focus on ethical responsibility, which I have mentioned much earlier in sort of the things that we're doing, not only looking introspectively and consistently or continuously into our operations and our partnerships, but also trying to promote thought leadership on ethical issues. Investment in research infrastructure, which is part of what we have all discussed earlier. We want to be able to create meaningful jobs. So some of the experiences we have found is that there are still quite a number of well-trained Africans out there that have studied in a number of other Western countries that are actually looking for opportunities to come back and create impact in Africa. Our modest contribution in sort of the expansions we are having is to enable that for as many as we can modestly do. Knowledge transfer is at the core of our work. Or what we are looking at ensuring that we work together with all our partners to be able to, from the data we generate through our partnerships, publish widely in order to be able to add to the body of knowledge that is already out there. Empowering African genomic researchers was one of the core areas that I had mentioned through the African Center of Genomics, Translational Genomics, but of course also through some of the work that Dr. Jumi is doing at the Genomics Molecular Genetics Institute. Environmental sustainability is at the core of what we're doing. And lastly, but not the least, is benefit sharing, which is also very critical and important to, some of the work that we're doing. We want to be able to ensure that within the framework that is available out there, we're able to give back to the societies that we're working in ways that demonstrate, first and foremost, back to the first issue that I mentioned here, our ethical responsibility to not just our participants, but the African society at large. Thank you very much.
And hello, everyone. My name is Delali Atipo. I am 54gene's Chief Operating Officer, and you have heard from our three esteemed colleagues today really on the tremendous work that we've been doing in partnership with the scientific community. And as we expand our work, we're quite excited to be able to come today to share a little bit around, and Amina pointed a little bit to this, a sneak preview on one of our exciting collaborations that we are doing with the Tanzania Human Genetics Organization and 54gene. And so it is my honor to be able to present our joint statement around our collaboration. The Tanzania Human Genetics Organization in Dar es Salaam and 54gene in Washington, D.C., United States, have on this day of the 3rd of September, 2021, mutually agreed to launch a joint collaboration. Both parties have expressed their desire to cooperate and pursue their common objectives in promoting genomics research and its impact on the health of Tanzanians and other African and global populations. With the launch of this joint collaboration, both parties will work together, respecting all extant regulations for ethical conduct in Tanzania, to implement two main projects. The first main project that we're going to be collaborating on is called the Tanzania Human Genome Variation Study. And the second one is a Tanzania Rare Disease Initiative. The goal of these two projects is to facilitate research, innovation, and technology transfer between our two organizations in various areas of human genetics. This endeavor will include, but is not limited to, applied science, technology, and innovation for the advancement of our common goals and joint generation of intellectual property. Really, we are quite excited for this collaboration. And as mentioned, always looking to further partnerships so that we all go far together, as we've noted and as Mina noted in the true African proverb, because we believe that we can all be able to advance genomics research further together, because we'll be able to grow further together, and we can't do it alone. So with that, I turn it back over to our moderators for the day, but we look forward to addressing any questions or comments towards the future as well.
Yeah, thank you very much, 54 Genes. A kind reminder to the attendees, this is a promotional presentation from our industry partner, and it's a pleasure to have this partner, future collaboration with 54 Genes for some of the support of the networking activity of the African Society of Human Genetics. We have in the chat room 2 questions and / comment. I think probably one of the questions been already answered. Is from Julie McCanny to advise on country you are in conversation with to explore synergies. One is Tanzania, we heard, and is there any other country that you are ready to disclose?
So not at this time, because by the nature, we want to be able to respect the partners that we're having those conversations and dialogue with. But we are definitely in conversations with a few other partners and trying to explore ways for which we can, as mentioned, further the research of respectively of both partners, but how do we, again, go further together? But if there are further questions or other organizations or individuals that are interested in being able to explore partnerships, we definitely welcome those conversations. But yes, in short, we are definitely having those conversations, but want to respect also our other partners that we're still in those conversations with. So won't mention at this time, but we definitely are very excited about the work with the Tanzanian team.
Okay, thank you very much, Achipo Delali. I will move to the chat. There is a lot of interest actually in question. The first one is from Dr. Mazandu at 54 Gene. The current 54 Gene storage capacity is 300,000 samples. How or when will 54 Gene be able to contain in the scaling process the 3 million African genome? I must say I did not consult with Dr. Mazandu at all. Yeah, when can we expect that capacity to be increased? The second question within the question, from data collection, management, storage, and to precision medicine, there is a huge gap, and how 54 Gene data science team will try to bridge this gap. So 2 questions. One around the data science, the second around the biorepository.
Yeah, I mean, I guess, you know, it kind of reflects the talk we heard earlier about, you know, as we continue to scale the data sets that we have, you know, will we run into major storage issues? Yeah, well, I think we're still thinking that through. We use S3 buckets, and we kind of, you know, all our data are, you know, stored in the cloud currently. But I think we will need to future-proof this and not just assume that the technology and compression will, you know, get much better over time. So yeah, we hope to continue our analysis and storage, you know, in the cloud, and use that as a framework for future analyses.
Okay, thank you very much. Yes.
Go ahead, Jimmy, and follow up after.
Thank you, Ambrose. I was just going to comment on the biorepository question. So the figure you've heard today isn't static. We review the storage availability as we go along, and we have very solid partnerships with cold storage providers to enable us to expand on cold storage, which is the primary way we store the majority of the samples that require cold storage. So it's what it is right now at 300,000. It's not to say that's what we're aiming for. ultimately would be looking to expand within one country, but also across multiple countries in Africa.
Yeah, thank you very much, Jumi. The next question is from Professor Ramsey, from Michelle Ramsey, on how does your GNA array, Genesis, compare to the H3 Africa array?
Yeah, it's smaller. It's about 800,000 markers. It's probably not as comprehensive overall. It's kind of we really wanted a kind of a nimble, low-cost array that would have a decent backbone. And so when we started running imputation, that it would perform pretty well. So it wasn't really about having functional content. It was more about having, a decent array for imputation. But there is some custom content there, and the custom content is a mixture of Nigerian-specific genomes and public available data sets, the latter being probably more relevant to Africa in general, namely the African EQTL content and GWAS catalog, African-specific significance. So there are kind of a number of differences. Probably the number of markers is the biggest.
Yeah, thank you, Colin. Yeah, additional comment from you, Atipe?
Yeah, no, I was just going to mention, and probably the additional bit to add to what Colin has mentioned is that the intention as well, too similar to what Jumi mentioned about our biobank, is that it's not meant to be static. We anticipate that we'll continue to add content over time to continue to increase the diversity of our Genesis chip as well.
Okay, thank you. Another question from Professor Ramsey. There is a question, there is a response here from Abasi from the chat, but it's probably a good opportunity to get anybody else to give some comment on it. Does the hospitals you work with in Nigeria have electronic health records? What data do you use for the FAWAS studies? Two questions actually. The one around the electronic health record in Nigeria. The second, the data you use for the FAWAS.
Yeah, just following from Basi's response, it's largely kind of questionnaire based. It's a pretty comprehensive questionnaire, but it's not ICD-10 level of detail. So, but we are actively looking into rolling out EMRs in specific partner hospitals, and so for FAWAS, I mean, you can.
Can you speak a little loudly?
Oh, I'm sorry. Yeah,
it's hard to hear you.
I'm sorry. Yeah, so for we can do FAWAS with the questionnaire data that we have, but it will benefit better once we have a more comprehensive specific hospitals.
Okay, another question from Professor Vale is how will your data contribute? Actually, David, you're on the stage. You can ask the question directly.
Yes, and I thought this discussion was very exciting, and the various tools you're developing and so forth. I'm wondering to what extent, to make optimal use of this, will you be able to support educational activities?
So, I mean, in terms of, just to clarify your question, David, so in terms of being able to support and partner with other researchers that can leverage ours. So yes, the short answer is yes. And Jumi touched on it a little bit earlier, but our intention is that, especially through what we're doing in collaboration through the ACTG that Aminu mentioned earlier, we want to be able to open up our doors for collaborations with researchers or grants that can be provided or services that can be provided to support individual research projects that might be going on within the scientific community. That's something that's very important to us, so that access can be there. So there's ways for which we've formalized that within and through ACTG, but then also, which is why we said we'd be open for folks, contacting for further discussion, but we do want to be able to provide that opportunity.
Terrific, thank you.
Yeah, thank you very much. I'll look for if there is any other question in the chat. A few congratulations for the the collaboration developed with the Tanzanian society, just more comment than question. We have 8 minutes at the top of the hour. Is there any final comment before we conclude from 54 Gene?
No, just that we look forward to, I mean, we've heard a lot of interesting talks today, and I know we have some further talks coming later. Just commend the society for the great work that's already, been ongoing, and we look forward to continuing to partner with other different folks across this platform. So thank you for the opportunity.
Yeah, thank you very much. Dave, any closing comment on education and training and future and possible collaboration, because we intend to maintain the collaboration with 54 Genes, specifically on the network and the educational activity. Maybe that may be part of the thing that we will do with the Bar Harbor. Any comment, Dave?
No, I just think I think the whole session has been very interesting. I'm not commenting on my own contribution, but I think the other presentation is very interesting, and so I too congratulate the African Society of Human Genetics on making this happen, and I'm very impressed with what 54 Gene is doing.
Okay, thanks very much. To all the presenter, Professor Mulder, to Professor David Valley, that's actually stepping without warning in place of Dr. Ray, to 54 Gene for your partnership and support for this meeting and for the following meetings we will be having. The next meeting we are having will be in Morocco between 12 and 18 months. So we hope that it will be an in-person meeting. We really pray that it will be an in-person meeting because the online platform is not conducive for some of the around the coffee conversation and negotiation. And for all the attendees on the platform, and we thank you very much for the interaction. A living session is always the best of the sessions. Keep connecting with the African society and keep collaborating and engaging with the African societies, particularly from the young investigator I can see in the room. Engage with the Young Investigator Forum, engage with the society activity, provide new ideas that will allow us to improve the operation and vision of the society. So we will have, after this session, 2 parallel sessions, one session on neurodevelopmental disorder. Please try to select which one you want to attend. A second session that we mostly focus on cancer genomics. So thank you very much. You will have 5 minutes before joining the next session at 1 p.m. Central African time. Thanks a lot.